So before we start and to carry on with the tradition of the Collège de France, I'm extremely pleased and honored to present and even to give this medal as an official acknowledgement of your outstanding lectures.
Oh, beautiful. Thank you.
As you can see, it is also a token of the gratitude of your enthusiastic audience. Okay, so now we will move to your last presentation. And today you will discuss cell adhesion, migration, and consent. Thank you.
Merci beaucoup, cher Professeur Petit, and ladies and gentlemen. It's just been an absolute pleasure for me to deliver these lectures in a place with so much tradition and establishment of excellence as the Collège de France, and it's been a wonderful experience for me, really an opportunity to not only have this experience to meet a number of you in the audience and also to visit and really get to know my wonderful colleagues in Paris and also in Strasbourg to a greater extent. So it's been a great experience for me. And I noticed a number of new faces as well as some familiar faces in the audience. So I will try to tie in the previous two lectures. And for those of you who weren't able to come to those, then I think you should be able to still gain something from this particular lecture. As those of you who have been to these lectures already know, I focus on skin as a model system for study, and that will be the tradition as well for today's lecture on cell adhesion, migration, and cancer. And for those of you who have been to the former two lectures, you'll know that I always like to start by choosing examples, and for 4 different lectures at the Collège de France and Strasbourg, I keep drawing upon a number of personal trips that I've taken to use these as examples of basically the diversity that exists in terms of our fascination with skin, not only nature's fascination with skin and her diversity with regards to various different body surfaces, but also we as human beings have managed to continue that tradition that nature has begun for us, trying to adorn upon what nature has given us. And today's lecture is going to focus very heavily on cell adhesion and the cytoskeleton in the skin epithelium. And I thought I would begin just by going through a few of the basics about skin and hair. The skin is the largest organ of our body, and it exists for a major purpose, and that is to regulate body temperature. And it does so by a whole repertoire of different tools. There are about 20 different cell types in the skin, and body temperature is regulated not only by the blood vessels, which are located in the dermis of the skin, but also in subcutaneous fat layer down here below the dermis. Sweat and oil glands are important in regulating body temperature, as is hair. And so there are a diversity of different structures within the skin that do this. Skin is also a sensory organ, and there are nerve endings that terminate within the epidermis. And in addition, there are Merkel cells which convey a sense of touch so that we can tell if our hand is on the stove, that we can remove it from that. And the skin is also very important with regards to protection from physical injury, and that's predominantly conveyed by the connective tissue within the dermis, composed of collagen fibers, elastin, resilient proteins that offer protection of the rest of the body organs. The skin is the largest immune system of our body. It contains Langerhans cells for antigen presentation located within the epidermis. There are specialized gamma delta T cells that are our first line of defense against infection. And then finally, the skin epithelium serves as a barrier to keep harmful microbes out and essential bodily fluids in, and that's conveyed by the epidermis, shown in brown here, and its notable appendage, the hair follicle. And if we didn't have epidermis, we'd basically dehydrate and die within about 24 hours of time. If we managed to survive the dehydration, we'd die of infection. So even though the epidermis is only composed of about cellophane thickness, and is a very small component of the skin, it's a very important one, and in fact an essential one. We use skin as a model system for studying cell biology, morphogenesis, and cancer because it's readily accessible, its lineages are spatially and temporally well defined. Adult skin epithelia undergo constant turnover and wound repair every 4 weeks period of time. You've got a brand new epidermis to your body surface. And skin epithelial stem cells can be maintained and propagated in culture, which really affords us mechanisms to dissect out the basic features of the skin epithelium and really understand at a molecular level how it's able to convey cell adhesion, migration, and how these processes go awry in cancer. And there are many different skin cancers that exist within the that are derived from epithelial cells of the skin, and as I pointed out over the previous lecture, they include some of the most prevalent cancers of the world, basal and squamous cell carcinomas. And toward the end of my talk, I'll focus on squamous cell carcinomas, which we haven't talked about with regards to the previous discussions on tumorigenesis thus far. So the focus of today's lecture is going to be on the epidermis. Last week we talked predominantly about the follicle stem cells and their ability to generate new follicles with the hair cycle, and also to regenerate epidermis and sebaceous glands on wound healing. Today we're going to be talking almost exclusively on the epidermis, and the points that you need to know here is that there are really 4 different stages of epidermal differentiation, and the fueling source for the epidermis is the basal layer of the epidermis. It's associated with an underlying basement membrane rich in extracellular matrix and growth factors, and this is the layer of dividing cells within the epidermis, and these cells are marked by their ability to adhere to the underlying substratum through 2 sets of integrins. One is a core of alpha-6 beta-4 integrin that forms structures that we call hemidesmosomes that interface with the intermediate filament cytoskeleton, and the other are the focal adhesions whose core is alpha-3 beta-1 integrin in the epidermis, and focal adhesions interface with the actin and microtubule cytoskeleton. Both of these are essential. Hemidesmosomes play the lion's share role of cell substratum adhesion. Focal adhesions are necessary in order for these cells to migrate and to fill in or move into a wound site and coordinate actin and microtubule dynamics that are important in cell division and a number of other essential properties of the epidermis. As these cells withdraw from the cell cycle and move outward toward the skin surface, they undergo a dramatic change in their program of transcription as they depart from the underlying basement membrane. In the basal epidermal cells, there are 2 positive-acting tyrosine kinase growth factor receptors that are very important, epidermal growth factor receptor and the insulin growth factor receptor. And then there's the TGF-beta receptor that has a negative impact on signaling in the epidermis. And we know from the studies long ago of Juan Massaguet that epidermal cells exposed to TGF-beta withdraw from the cell cycle. It's playing a negative role on putting the brakes. It basically puts the brakes on the growth of the epidermal keratinocyte. All of these genes are downregulated as cells commit to terminally differentiate. In the basal layer, there are predominantly intercellular junctions composed of that are adherence junctions, as opposed to desmosomes, which are relatively sparse in the basal layer. Keratins 5 and 14 are the keratins that are associated with not only the proliferative cells of the epidermis, but also proliferative cells of virtually all stratified squamous epithelia and a number of glandular epithelia. And as these cells differentiate, they switch off this program of gene expression and they switch on new transcription factors. AP2 is one that we've studied, CEBP. NOTCH signaling is upregulated at this transition. HES1 is a transcription factor that is under the control of NOTCH signaling. It is induced at this juncture. And in addition, now there are many more desmosomes and now a new set of keratin filaments composed of keratin 1 and keratin 10 that are switched on as these cells commit to terminally differentiate. And in the spinous layer, these cells really adopt a feature of a robust keratin network linked extensively through desmosomes. As these cells continue to move outward, they undergo still changes at the transcriptional level. Even though the cells left dividing, stop proliferating in the basal layer, they still transcribe genes. The new genes that are of particular interest are loricrin, which is a protein that, along with a number of other proteins, are deposited underneath the plasma membrane of these cells. Loricrin is typified by having a very abundant number of glutamine residues and abundant number of lysine residues, and that comes into play as these cells move outward toward the stratum corneum, become dead cells, because at that time there's an influx of calcium activating transglutaminase. Transglutaminase gives gamma-glutamyl lysine cross-linkages that make an interconnected, essentially cornified envelope that serves as an indestructible sac to which these keratin filaments then are organized in. In addition, profilaggrin, a precursor of filaggrin, is transcribed at this stage, and it's processed to filaggrin at the transition at the end. And when it's processed to filaggrin, it serves as a glue to make cables of keratin 1, keratin 10 filaments that basically are also indestructible. So the idea is that by the time these cells reach the surface of the skin, they're absolutely indestructible. And at this stage, they also undergo an apoptotic-mediated-like mechanism that involves caspases and many of the traditional players. It also results in the enucleation and destruction of all the cellular organelles as these cells become effectively dead sacs, proteinaceous sacs. Then one last note, and that is that there are granules called lamellar granules or lamellar bodies that start to accumulate in the granular layer, and at the ultrastructural level, these granules are packed with lipid bilayers. Those lipid bilayers protrude and extrude along the surface of the granular cells into the stratum corneum and provide basically lipid seals that keep our body's surface so protected against bacterial infection and against water loss. And that really is the whole intent of this entire process of terminal differentiation. These dead cells will eventually be sloughed from the surface, continually being replaced by inner layer cells moving outward. That's the process of homeostasis in the epidermis, and we'll get to how this homeostatic process has to change as there is a wound induced in the skin and the cells have to continue and move in a different direction and fill in, proliferate and fill in at the wound site. So, given what I've told you thus far, effectively the keratin filaments of the epidermis are to the epidermis what globin is to a red cell, and it made perfect sense some many years ago now when I was a postdoctoral fellow and interested in studying the epidermis and epidermal biology for my future career to focus on keratins to begin to give us markers, molecular markers, that would allow us to study this process of epidermal differentiation and to learn about the dynamics of the way in which this tissue can really undergo wound repair, and how, when the processes go awry, cancer develops. And so we started to focus on these keratins, and back when I was a postdoctoral fellow, learned that keratin 5 and keratin 14 mark the basal layer, as I've told you. The keratin 1 and keratin 10 were induced at the level of messenger RNA and transcription, and as cells committed to terminally differentiate. We also studied keratin 6 and 16, which are induced suprabasally on a wound response, and as Henry Sun and his co-workers showed, are also expressed aberrantly in a number of different hyperproliferative disorders of the skin. There are hair keratin proteins that provide or produce the hair shaft cells, and I spoke about those last week as direct downstream targets for Wnt signaling. All of the promoters for all of these genes, as I later showed when my laboratory began at the University of Chicago back in the early 80s, are indeed regulated by different sets of transcription factors, imparting to them this difference in programs of gene expression. And what's beautiful about this system is that these structural proteins and their genes are basically serving as marks of particular stages of differentiation and behavior of the cells. And that's been useful and utilized not only by my laboratory, but by many different labs over the years. I just point out that the keratins, even though they are extremely diverse within the skin epithelium and really take their place in the skin as being major players, that there are keratins in all epithelial tissues of the body. And as Werner Franke studied extensively, keratins 8 and 18 mark the simple epithelial keratins of internal tissues such as intestinal or colonic epithelium. So when I began then at the University of Chicago, we started by making a cDNA library, cloning and characterizing the keratin proteins, studying their sequences, determining their sequences, using secondary structure predictions to learn that these are large coiled proteins. And we then began to look at what is the structure and what is the function of these proteins. And what we learned in raising antibodies to the keratins and to the proteins that associate with keratins, in this case marked by a desmosomal protein, you can see this beautiful extensive network in primary epidermal cells grown in the laboratory, and you can see that there is a very clear-cut connection between the intermediate filament cytoskeleton and sites of cell-cell interactions, and I'll get to that in a moment. We also learned that the basic subunit structure of keratin is an obligatory heterodimer, hence explaining the program of gene expression that I had identified, where keratins were typically expressed as pairs of type 1 and type 2 proteins. We showed this at the biochemical level, and also worked out some of the steps along with a number of notable figures, amongst them Klaus Weber, in terms of elucidating the overall structure of the 10 nanometer keratin filament. So each keratin filament is comprised of about 10,000 to 20,000 individual keratin proteins. As we now had all of these different keratins cloned, we began to identify residues which were important in this filament assembly process. We had an in vitro filament assembly process and an in vivo one, that is to introduce keratin proteins into the primary keratinocytes using gene transfection. And here's an example, a very early example, where an epitope tagged wild-type keratin protein integrates into the existing keratin network. And in this case, a single point mutation strategically placed in an area that we had identified as being critical for the assembly process. That single point mutation integrated into the keratin network and completely disrupted the keratin network, leaving clumps or aggregates of keratin protein in the cytoplasm. And at this stage in my career, we began to wonder these are the major structural proteins of the skin. We had identified mutations which acted in a dominant negative fashion that, when introduced into cells, caused complete disruption of the keratin network. What is this network doing, and how might it relate to human genetic disease? And this really started my interactions with Christine many years ago, and our discussions at genetics meetings, because at this point we meaning my graduate student and I, basically left the University of Chicago momentarily to be able to learn how to do transgenic mouse technology. It was just coming on the scene at the time, and amongst the first transgenic papers that we worked on was basically to take our mutant keratin and introduce it into mice and ask what happens when this dominant negative acting keratin 14 mutation is basically expressed in the skin of the mouse. So we used our keratin 14 promoter and the keratin 14 mutation, and the results were striking. And that is that initially the mouse looked okay, its skin looked okay, but as soon as the mouse started to move around in the cage, it started to develop blisters, in this case on the paw regions, and those blisters were manifested further. When we began to look at how the blistering arises, what we found is that the blistering arises for a very interesting reason, and that is that it's not simply a separation of the epidermis, as it might look like, from the underlying dermis here, but it's actually a rupturing of the basal epidermal cells expressing this keratin network. And effectively what we had generated is an epidermal basal layer that acts like eggshells. As long as you don't touch those cells, they're fine. But as soon as you apply mechanical stress to the surface of the skin, then those cells burst or rupture under that mechanical stress. They cannot withstand the mechanical stress in the absence of a proper keratin filament network. And so we then bought a dermatology textbook. I'm trained as a PhD, not an MD, and we fortunately diagnosed these mice correctly as having a disorder, epidermolysis bullosa simplex. It's a human blistering disorder affecting about 1 in 40,000 in the population. And this blistering disorder can be very severe. And in this case, this child simply had his face washed, and that caused the blistering. So very mild trauma in the severest cases can cause blistering. And it's obviously not only a physiological damaging, but also psychologically damaging disorder. Skin disorders have that tendency, unfortunately, because it's right at the surface and we can see every disorder. So it's a gold mine for a human geneticist, gold mine for an armchair geneticist like me, but it clearly is psychologically damaging for those humans that suffer from various skin disorders. So we then began to look at what happens with milder keratin mutations, and what we learned in the course of our studies is that there were 3 different human genetic diseases that all fell into this class. The etiology of none of these diseases had been worked out, and what we demonstrated is that all 3 of these diseases in humans are indeed due to mutations in keratin 5 and keratin 14, and that it is the degree to which one has mutations that perturb the overall filament assembly process that determine the severity of the blistering disorder. So there was a direct correlation between the biology that we had been doing for 10 years previous to this and all of the physiology that dermatologists had identified and characterized for this human disease or this group of human diseases. This was also the first human disease known of an intermediate filament protein, and we then began to become very interested in human genetics, and the first disorder that we veered from was this one. It's known as epidermolytic hyperkeratosis, where patients have a crusting of their skin, and it's typically over flexural regions, in this case the knees and the elbows, areas again of mechanical stress. And at this ultrastructural level, there were clumps or aggregates of keratin protein or keratin-like material in the suprabasal cells. And since as a postdoc I had identified this change in gene expression that occurs as cells undergo a commitment to terminal differentiation, it seemed like an obvious leap of not too much faith to predict that epidermolytic hyperkeratosis was going to be a human disorder of keratin 1 and 10, which we immediately focused on. And this time, instead of taking 10 years to figure out the genetic basis of a human disease, it took us less than a year to work out this genetic basis. And at that point, you could make a whole list based on differential patterns of gene expression that we and others had worked out over the years, and a whole list of different potential human diseases that matched this paradigm where cells would undergo cytolysis or rupturing upon physical stress, undergo degeneration, and at the ultrastructural level had clumps or aggregates of material that had the same electron density as an intermediate filament protein. And so at this point, not only could we predict a long list of potential human diseases of keratin, but also a whole number of other people in the world could predict the same. And at that point, we decided to shift from looking at epidermal diseases and keratin disorders, which were fairly straightforward, and say, could we test this paradigm further by looking at other intermediate filament disorders or potential intermediate filament disorders. So we teamed up with a group in Spain that then had been studying a human disorder of a generalized myopathy that where patients had died of heart disease at about 20 years of age. It was a rare form of a generalized myopathy, and Klaus Weber and his co-workers had studied desmin for many years and demonstrated that desmin was an intermediate filament protein expressed in all muscle cells, and that in fact the desmin protein localized to Z-bands of muscle cells. And we soon then focused on this particular group of rare patients where there were clumps or aggregates of protein with desmin intermediate filament-like electron density at the ultrastructural level, and worked out that those patients had mutations in desmin that we showed were functionally responsible. At that point, we left the intermediate filament world and in fact have not been back to it since then, and yet this is the list of various different intermediate filament diseases in the year 2008. We began to focus on architecture, cytoarchitecture in a cell. The notion that intermediate filaments aren't simply bags, the cell's not simply a bag of filaments, but these filaments have architecture, they have structure, they're adhering to desmosomes at intercellular contacts, they're adhering to hemidesmosomes. What are the proteins responsible for making those connections, and what's the physiological relevance of those connections? So this then led us to the genetic basis, this time of a well-known neurological mouse mutant that had been discovered back in the 1960s, whose genetic basis was unknown, where the mice had mild skin blistering but severe sensory nervous system degeneration. We went on to demonstrate it's due to a cytoskeletal cross-linking protein that links the intermediate filaments to integrins, but this protein was only known to be expressed in the epidermis, and this then opened the door for our studies of this first protein, which is a spectroplakin family member, now a new name for this class of proteins. It turned out that this is a huge gene encoding many different exons, and the intermediate filament exon was present in both neurons and epidermal cells, but the integrin-mediated connector exon was not. It was spliced out in the neural form, and spliced in was an actin binding domain and a microtubule binding domain, and there are now about 4 different variations that are expressed in the nervous system, and some of them don't even have connections to the intermediate filament network. So this really changed the focus of my lab to now becoming much more interested in the actin cytoskeleton and the microtubule cytoskeleton. It also gave us a real interest in looking at interfaces at junctions and cytoskeletal interfaces. It also led to the genetic basis, not by my laboratory, but by others, of a human disease that was a complex disease between EBS and muscular dystrophy that had an obvious connection because these proteins are related to one another and fall into the same family. There are now additional various different human genetic diseases that involve cytoskeletal linkages to either integrins or cell-cell junctions. One of them is a disorder that is known as striate palmoplantar keratoderma that's associated with severe cardiomyopathy. And these particular patients have blistering, and that blistering turns out to be due to the rupturing to mutations in the desmoplakin protein, which links intermediate filaments to desmosomes. And the C-terminus appears to be very important, which was interesting because both Kathy Green's laboratory and my laboratory had identified this domain as being a domain that is important in linking to the intermediate filaments. So to summarize, 20 years or 15 years of research in my lab and other labs that the intermediate filaments and desmosomes are prominent in tissues subjected to a great deal of mechanical and physical stress, which are predominantly the epidermis of the skin, and the heart muscle forms a robust infrastructure to the cell, and when defective leads to mechanical disorders, fragility, blistering, rupturing of cells upon mechanical stress. So today's title dealt with cancer, not with degenerative diseases, and that really then provides me with the segue to starting to focus on what's the importance of having cell-cell junctions and cell substratum junctions that not only link to intermediate filament networks, but an entirely separate network that links to the actin and the microtubule cytoskeleton. And so we started to look at the importance of the actin cytoskeleton, and this led us into the classical cadherins. And just to give you an indication of how they differ from desmosomal cadherins, they're members of the same family. It's their cytoplasmic domains that have subtle differences that serve as platforms for the interaction of a group of related proteins, beta-catenin in association with classical cadherins and plakoglobin in association with desmosomal cadherins. And plakoglobin links to desmoplakin in the intermediate filament network, while beta-catenin links to alpha-catenin, an unrelated catenin protein, which then recruits proteins as well as linking to the actin cytoskeletal network. P120-catenin is a relative of these two and binds upstream from beta-catenin. So we've used genetic engineering, initially transgenic work that I showed you for keratins, but very soon after that, knockouts and conditional knockouts using our keratin 14 promoters and keratin 5 promoters to target the expression of Cre recombinases and inducible Cre recombinases. And these are amongst the genes for these particular junctions that we've targeted for ablation over the last 10 years or so. With regards to adherence junctions, we learned very early that one of the importances of these particular junctions is to draw cells together that are physically apart from one another. The actin cytoskeleton becomes central in that. And here one can see that there's a very characteristic a series of puncta that are marked by cadherin molecules, classical cadherin molecules. And in this case, it's in response to taking primary epidermal cells and adding calcium. That stimulates both classical adherence junctions as well as desmosomal junctions to form. And you can see that in time progression, this adhesion zipper, as we called it at the time, really zips up to leave a continuous sheet of sealed membranes here by about 20 hours after we've added calcium to these cells. So when we began to look at the relation to actin, shown in red here, we could see that not only were there these radial cables of actin, but in fact it looked like actin was extracellular. We could see actin here, and that turned out to be due to filopodial connections that bridge these two cells and that clearly interface with these puncta of adherence junctions. And we then used our conditional targeting to ablate alpha-catenin and realized that in the absence of alpha-catenin, not only did we lose the ability to form these adherence junction structures, but in fact, we also lost the overall organization of the actin cytoskeleton. That was perturbed as a consequence of the loss of alpha-catenin. And there were several very interesting papers that started to emerge at this time. One of them from Wood and Martin, who were looking at dorsal closure, a process of bringing two epidermal sheets together in the fly embryo. And interestingly, in flies that were expressing GFP actin, they showed many of the same features, these radial actin cables in areas where junctions were sealed versus these filopodial-like extensions out into the areas where the sheets were still closing upon one another. And Alec Baezi, a graduate student in my laboratory at the time, engineered GFP actin mice expressing about 2 to 3% of the wild-type levels of actin, not enough to perturb in any way the actin dynamics within the animal and within the epidermal cells, but enough to give us beautiful images here. And here we've co-stained with antibodies to myosin, and you can see that the bridges, the filopodial bridges that form connections between the cells in the initial stages of intracellular adhesion are not marked by myosin, but there's an underlying myosin sheath. And so it's really a matter of using the actin-myosin cytoskeleton to move the cells together, to bring the cells together and allow them to seal and make a continuous epidermal sheet. And that's a very important process as you learn from the earlier schematics about what epidermal cells must do. So here we've used video microscopy, taking skin and the beauty of cell culture from the skin directly of these animals. And here are keratinocytes that you can see now forming these puncta and their coordinated radial cables of actin. And you'll see in a minute that it becomes very obvious what adherence junctions are doing to generate an epidermal sheet. Not only do they provide the initial stages of bringing these cells and organizing these cells together in a sheet, but what they really do is to basically integrate the entire actin cytoskeleton across the epidermal sheet so that it's behaving as one large cell with one large cytoplasm or cytoskeleton. And you can really almost visualize cables of actin going all the way across the sheet, that these are linked by virtue of the adherence junctions one to the other. And that's really what is critical with regards to the behavior of these adherence junctions. So it's really coordinating the actin cytoskeleton so that a sheet of epidermal cells can act as a unit and behave as a unit, something that tissues do, something that a yeast cell sitting in a culture dish doesn't have to do, or an isolated cell sitting in a culture dish doesn't have to do. Clearly something that is necessary for sheets to form. And this movie over here that you saw was that of what happens when you mate the alpha-catenin conditional knockout mouse on the background of the GFP actin actin transgenic mouse. And what you can see is that when you don't have alpha-catenin and you cannot form stable cell-cell junctions, you also don't have any communication or coordination with the actin cytoskeleton across an epidermal group of epidermal cells. So even though these cells are closely juxtapositioned to one another, there's membrane ruffling in all different directions that continues to go on. The actin cytoskeletons for each of these cells are behaving independently. So to get cells to act as a tissue, as a coordinated tissue, they have to have these types of junctions. So keratinocytes function as a sheet through adherence junction and actin dynamics. And presumably they also function in 3 dimensions using these kinds of systems. And we're currently in the process of just starting to do in vivo live imaging on embryos to be able to visualize the 3-dimensional space and architecture of the cytoskeleton. And that's something that we're very interested in doing. At each step along the way, expanding our repertoire of knowledge and expanding the tools that we use to be able to look at this. So by now, I think you're convinced of the coordinate action between adherence junctions in the actin cytoskeleton, but how does alpha-catenin recruit the actin cytoskeleton, and why is it so essential? Well, this has been a matter of debate and recent debate, and that debate still continues about exactly how that happens. But the field is increasingly understanding more about the process. James Nelson and his co-workers are excellent biochemists and have done some beautiful biochemistry work over the years on this problem. And what they've shown recently is that in vitro, alpha-catenin does bind to actin, and in fact that's something that's been known from Rim's work many, many years ago. But it turns out that they do so predominantly as homodimers, at least in vitro, and as homodimers they can bind to F-actin, but as heterodimers complex with beta-catenin at an adherence junction they cannot. And so this is where we see radial cables of actin, whereas it looks like the binding of alpha-catenin to actin is happening independently of the adherence junction. And so one model that you could come up with as far as how this might happen comes from an additional observation in cell culture, and that is that, or in a test tube, and that is that at about 30-fold higher than physiological concentration, we don't know whether this is physiologically relevant, James showed that alpha-catenin homodimers inhibit ARP2/3 mediated actin polymerization. This is a polymerization process that creates branched actin filaments at those lamellipodia, those membrane ruffles that you saw in the movies. And what you may have remembered from the movies is that as cells came together, they dampened out the amount of membrane ruffling and membrane activity that was occurring. So as cells formed stable cell-cell junctions, they dampened down that process. And so you might argue, and in fact a reasonable model that is consistent with these hypotheses and these studies, is that what happens is that as you accumulate adherence junctions at membranes, you raise the local concentration of all of these proteins, and as you raise the local concentration of alpha-catenin at membranes, you might start to get this kind of equilibrium going on, such that you now have alpha-catenin homodimers forming, which can then bind and inhibit the overall process of lamellipodia as junctions seal the membranes. And that's a hypothesis that is in a working model that's basically consistent with all of the data at hand. Whether it's demonstratable as an in vivo entity is something that remains to be demonstrated. So if alpha-catenin associates with actin independently of its ability to form adherence junctions, or at least maybe dependently, but indirectly, then what does organize actin cables at adherence junctions to have the actin coordinated across an epidermal sheet? Well, there are many other players involved. In fact, there are right now too many players involved to explain that hypothesis, to explain that unanswered question. And one of them is a protein called VASP, which for many years now has been demonstrated to act as a elongator of actin filaments, and it does so by competing for the barbed capped end of a dormant actin filament, and then being able to elongate actin as a consequence. And VASP is a protein that we showed when we knocked out alpha-catenin to no longer associate with sites of intercellular junctions or at the plasma membrane, so that in the absence of alpha-catenin, VASP cannot be recruited. In addition, by yeast-2 hybrid screening, we showed that alpha-catenin directly interacts with formin 4, and formin is a protein that we showed, the mammalian formin, to act as a nucleator to promote actin polymerization. It was already demonstrated that a yeast formin could do this, and Agnes Kobalak in my laboratory showed that in fact this mammalian formin can do the same thing. So actin polymerization could be mediated directly by formin 4. We don't yet know how VASP associates with classical cadherin complexes, but we do know that Takeichi and his co-workers looked at a related cadherin molecule called FAT1 that also associates with adherence junctions and that does interact directly with VASP. So there are mediated mechanisms by both VASP through FAT1 and E-cadherin complexes and alpha-catenin and formins that could explain how we get elongated radial actin cables that coordinate the actin cytoskeleton across a sheet. And in addition, there's work from Takai's laboratory that demonstrates that the nectin-afadin complex can also serve to integrate the actin cytoskeleton at sites of adherence junctions. So there are at least 3 potential models that could explain how actin gets there. We see that actin gets there, and assuming that James Nelson's studies are right, it appears to get there indirectly. Once membranes are sealed, they have alternating desmosomes, which are these railroad track-like structures at the ultrastructural level that are separated by adherence junctions, the electron light structures that you see here. And we vision that the desmosomes are acting as molecular clamps in the process of regulating cell-cell adhesion. So the model is that there are small GTPases then that stimulate the initial extensions of filopodia and lamellipodia that bring cells together, and then once cells begin to be brought together, making extensive defunct contacts over time, eventually adherence junction puncta form. Once that happens, we rapidly see the assembly into adhesion zippers. Desmosomes appear to be clamping that process in place to eventually allow the membranes to seal, dampen down then the GTPases, and shut off the lamellipodial and filopodial extensions in the cell. So now there's a link between cancers and adherence junction proteins that has been really established now since the early 1990s, when Takeichi's group was studying a human lung tumor cell that lacked alpha-catenin and showed that if you add back alpha-catenin, that you get cell-cell junction formation. So this set off a long list of studies from a whole variety of different groups associating or linking adherence junction proteins to cancer, and we've contributed to that with our work on the loss of alpha-catenin. And in fact, here you can see now an explant missing alpha-catenin, and these cells are really totally doing their own thing. In fact, you see multinucleate cells that also form for the explant, and here you see the normal directed migration of wild-type skin out of an explant. So these cells really almost look like cancer cells, only in contrast to cancer, which is generally mutations acquired over many years of time. This is tissue that's taken a day or two after ablation of alpha-catenin. So how does that become important? Well, the prevailing notion of why you need to dampen down junctional formation in cancer is that you've got all these various different mutations in classical tyrosine kinase pathways that over time give rise to an increase in RAS-MAP kinase activity and an elevated cell cycle. And then all you need to do is dampen down adhesion, and you've got this tumorigenic cell that can now invade and metastasize and move to other parts of the body. Well, there was some evidence at the time that suggests that that in fact may be the case by virtue of the fact that if you raise the level of tyrosine kinase activity, you can start to get phosphorylation of beta-catenin, and phosphorylated beta-catenin is no longer able to make as efficient junctions as wild-type beta-catenin. So this would be consistent with the notion. The problem is that when we began to look at alpha-catenin null skin, within a day or two after ablating alpha-catenin, now we don't have mutations, accumulated mutations in the RAS-MAP kinase pathway, but we still see hyperproliferation very soon on. In fact, when the animal is still an embryo. We also see sustained activation of the RAS-MAP kinase pathway, both at the immunohistochemistry level and also at the biochemical level. There's elevated RAS and there's elevated MAP kinase. And yet we haven't mutated the tyrosine kinase growth factor receptor pathway. Now we're indirectly, by virtue of knocking out an adherence junction protein, activating the tyrosine kinase pathway. That doesn't fit the paradigm or fit the data. And then you could say, well, maybe it just has something to do with cell-cell adhesion. So then we started to look at the cells in culture where we can reduce the level of calcium so that normal wild-type cells can't adhere to one another. We now no longer have adherence junctions, and we still see hyperproliferation in the loss of alpha-catenin. So that doesn't fit the paradigm at all for how cancer was envisioned at the time and the role for reduction in cell-cell adhesion. And so what's going on here? Well, now there's 3 models that we can come up with that could explain the data. And there's evidence, we have evidence to support all 3, although we have the most evidence to support the first model, this one. And that is that when you miss alpha-catenin, that there could be new interactions between E-cadherin and beta-catenin with other transmembrane proteins, including the tyrosine kinase receptor pathway and or downstream members. And in this case, we showed a direct interaction with insulin IRS1, a downstream effector for the insulin growth factor receptor pathway. That could explain the elevated levels of RAS-MAP kinase and sustained cell cycle elevation that we see. But another model could be that in the absence of alpha-catenin, there's interactions of these receptor pathway members now with integrins, which also have tyrosine kinases associated with them, non-transmembrane, but SARC and also FAC, and that superactivation as an aberrant interaction of these protein complexes could then explain the data. Then you could also surmise that in the absence of alpha-catenin, perhaps beta-catenin goes into the nucleus where it could interact with left TCF family members to evade the Wnt signal necessary for the activation of these target genes. And as you learned last week, some of those target genes are cell cycle proliferation genes. So we do see low levels of elevated Wnt signaling, and we do see some interesting aspects that could be explained through integrin-activated mediated mechanisms. And as I said, for now, we also see suggested evidence of downstream interactions with the tyrosine kinase receptors as potential means of explaining the tumorigenesis. But it's not so simple as we initially imagined the hypothesis based on what people knew at the time. We've also linked the loss of alpha-catenin directly to tumorigenesis. Those animals are not very healthy with their entire body missing alpha-catenin at their body surface, given what I've told you about the functions of the epidermis. But we can graft the skin, small samples of the skin, onto the backs of nude mice, as you learned last week. And when we do that, at almost exactly 40 days of every single graft, if we take a look at the skin, it looks almost exactly the same, and that is that we see these large internalized aggregates of tumor cells, very highly proliferative, marked by proliferating antigens, very low levels of E-cadherin at the membrane, and these look virtually indistinguishable from low-grade squamous cell carcinoma. By 70 days after the graft, we see epithelial mesenchymal transitions and a move to high-grade or reduced differentiated squamous cell carcinomas. In addition, we see inflammation all around the tumors that form. And in human squamous cell carcinomas, there's basically a very similar pathology. High levels of inflammation surrounding the invasive squamous cell carcinoma. So is this linked? You'd say alpha-ketinin already has enough functions. We don't need any more functions for alpha-ketinin. But in fact, alpha-ketinin links directly to the NF-kappa B pathway of transcriptional regulation of a pro-inflammatory response. And it does so very soon after we've ablated alpha-ketinin. We see signs of activated nuclear NF-kappa B, and we see signs of activated downstream targets of NF-kappa B, biochemically and immunohistologically. In addition, if that weren't enough, in the last year, we've collaborated with Al Reynolds and his co-workers, and we've ablated the P120-ketinin in the skin of these animals. And what we see is, again, a pro-inflammatory response that occurs whether we knock out P120-ketinin or alpha-ketinin, and we see it very soon after we've knocked out these genes. We've also looked biochemically, and what we know is that at the root of this appears to be the small GTPases, and when these adherence junction proteins are missing, then we see a superactivation of I-kappa-kappa kinase, which is necessary to phosphorylate and inhibit the inhibitor for NF-kappa B, which leads to NF-kappa B activation and inflammation. And so now we're starting to explain many of the features associated with squamous cell carcinomas of the skin, but these were not many of the features that one would have predicted for an uncontrolled or absent adherence junction protein, and again, against the prevailing view of how these various different responses occur. So if you recall from last week, we talked about the notion that the very mechanisms that we were studying that control stem cell activation, normal stem cell activation in the hair follicle, when mutated, gives rise to different types of cancers. And just as we took that avenue in that direction, we can take the reverse avenue, that perhaps the very mechanisms that are giving us cancers could be a reflection of a natural process gone awry. So what might be the natural process of having cell-cell junction proteins linked to proliferative responses, linked to an immune system response in the skin? Well, the natural system, again, requires a model, since we don't yet know exactly what's going on, but a working model always helps us to at least conceptualize what might be going on. And we think what might be going on is that cell-cell junctions linked to the actin and microtubule cytoskeleton, which is necessary to control cell division, might be there for a reason to control effectively or monitor the density of cells that exist within a tissue. How is that process controlled? If you've got cell-cell junctions monitoring density of cells within a tissue, then at low density, such as what occurs when the animal is rapidly expanding and you need to have a very hyperproliferative epidermis, if you have very few adherence junctions, which you actually do in embryogenesis, then you could imagine that you ratchet up the level of cell proliferation. That increases the cell density. As you start to produce a mature epidermis, now there's no reason to have such hyperproliferation going on, and that leaves us with an explanation for why we're controlling the immune response in this system. But let's consider what happens in a wound site. Now at a wound site, we've completely ablated all junctions at that site of the wound. We've created a gap. You certainly need to recruit a growth response for the cells to grow in and fill in at the wound site. But now there's a real reason for why you'd want to activate the immune system so that you can fight infection locally at the site of the wound. And now when the entire system is repaired and the cells are now adhering to one another, you'd want to dampen out that system locally. And when you don't dampen it out, or if you mutate any one of these junction proteins, now you start to get a constitutive cycle, as we see in squamous cell carcinomas. It's a working model, but I think one for which there might be a lot of rationale underneath. So I'm going to tell you, if you've got the energy, for a few final issues that I think raise an additional set of questions, and that is, we're dealing here with the basal layer of the epidermis, and this layer of the epidermis is the proliferative layer of the epidermis. And one thing that we noticed is that in the absence of alpha-ketinin, where there is normally a single layer of dividing cells, as marked in red here, there are multiple layers of dividing cells in the alpha-ketinin null skin. So what's controlling that process? And in the last few years, we've been really interested in how cells stratify and how they differentiate, and how this process might be linked to adherence junctions. And what we noticed more than 10 years ago is that in the embryo, when skin begins as a single layer epithelium and then starts to stratify, that we see at the light microscopy level a shift in the orientation of the spindle plane. And we couldn't explain that. Why would that happen? And we had no molecular explanation until Terry Lechler came to my laboratory as a postdoctoral fellow from Rong Li's laboratory, where he was studying the polarity of spindle and how the dividing nuclei get into the yeast bud. How does that nucleus enter the yeast bud? What are the dynamics of that process? And he took an interest in this problem in the skin, and he made a GFP centrin mouse that allowed us to monitor spindle orientation in the skin. And to make a long story short, it turns out that you can monitor this very nicely, and at 12.5 days of embryonic development, the spindles are oriented parallel relative to the underlying basement membrane. And at 15.5 days of development, we see this shift. And he also made a GFP ensconced mouse, which allows us, using a microtubule binding protein, to image the microtubule network now, just as we imaged and I described for you the actin network. And here you can see some of these asymmetric divisions relative to the underlying basement membrane. And in fact, if you do the statistics, what you see is that the majority start out parallel and the majority end up perpendicular or at a skew relative to the basement membrane. And this is not simply something that happens in the epidermis. It happens in other stratified epithelial tissues, and it's concomitant with the stratification of the tissue. So Terry looked at genes that could be involved in this process, and here we again take a page from fly genetics, and the fly neurobiologists had identified a group of genes that are genetically involved in asymmetric divisions in the neuroblast. We cloned out the remaining protein in mouse, the mouse inscutable, and it turns out that these proteins are in the right place and time to play a role. So this is par 3, and we see the same for APKC in the mouse, where this set of proteins is localized apically relative to the basement membrane in the dividing layer of the epidermis. What's known is that these proteins set the scene for the organization of inscutable and LGN, or in the fly, bazooka inscrutable and partner of inscrutable. And here, in these asymmetric divisions, we see that only in cells that are going to undergo asymmetric divisions and only cells that have entered mitosis do we see either inscrutable or LGN forming this cortical localization. So these proteins are there in all of the cells. These proteins are only there as cells enter mitosis and are going to undergo an asymmetric division. We don't see them at any other time, suggesting that they could be playing a similar role in the epidermis as they do in the fly. But right now, we have no genetic evidence. We're collaborating with Jürgen Noblecht to be able to look at this. So, what governs the process? Here we have a whole bank of different mutant mice to look at. So we looked at our mice lacking beta-4 integrin and lacking beta-1 integrin. And what we see is that when the mice lack beta-4 integrin, they still form the apical domain. In the absence of beta-1 integrin, now the domain is localized, but it's random, and that randomizes spindle orientation as well. And in the absence of alpha-catenin, now the cortical domain doesn't even form, and in the presence of loss of desmoplakin, they do. Spindle is randomized in the absence of alpha-catenin. So that starts to give us an understanding of why we see the dividing cells in the suprabasal layer. And in fact, in alpha-catenin, there's a loss or a defect in terminal differentiation as well. We've also linked a protein pneuma, which is a coiled protein, to that process. And this coiled protein binds to the microtubule cytoskeleton, suggesting a way in which the astral microtubules might be linked to this cortical actin cytoskeleton. And it's really the actin cytoskeleton that sets up this interaction of PAR3 and APKC and positions it apically. So we now start to have new insights into organization, not simply just of the actin cytoskeleton, but also interdependence with the microtubule cytoskeleton and also interdependence with the basement membrane. And why would the epidermis do this? Well, interestingly, it conveniently sets up the opportunity of how you'd actually maintain all of the proliferative components of the cell in the basal layer of the epidermis through one of these asymmetric divisions, because the integrins and the tyrosine kinase growth factor receptors are down here where their ligands are associated. And it then provides a paucity of those factors up here, and it also provides a potential platform for how you might end up then with various different factors associated with differentiation that could be associated in this compartment. Interestingly, knot signaling we know takes place in the suprabasal layer, and in flies there is an inhibitor of knot signaling that positions itself in the opposite domain of where inscrutable and partner of inscrutable are, suggesting again a possible mechanism which is obviously testable in our system. So we're really viewing then homeostasis as a very complex process of having symmetric divisions, asymmetric divisions, and Philip Jones and his co-workers just in the last 6 months in Cambridge suggested an even added complexity, and that is that in some situations there may be asymmetric divisions that occur basally, leaving both the committed cell and the uncommitted cell in the basal layer. And it really is suggestive that the whole process of homeostasis, one, likely has a number of backup mechanisms to make sure that it's maintained, and secondly, likely is a complex process that must be regulated based on the need for proliferation, more proliferation in certain instances, such as epidermal expansion or a wound healing process, versus less proliferation in perhaps an aging individual where homeostasis has really slowed down. So the last frontier then, dealing with migration, and dealing with backup mechanisms we've really revealed only in the last year, and that deals with the loss of TGF-beta receptor, which I alluded to last week, where I mentioned that the TGF-beta receptor, when we conditionally ablate it, really doesn't have much of an effect on the stem cell compartment in the hair follicle, but it does have a huge effect in the skin epidermal stem cell compartment, the basal layer of the epidermis. And you wouldn't ever know it, because when you look at the epidermis, it looks perfectly normal in the absence of TGF-beta receptor. And yet, remember, I told you that TGF-beta receptor applies the brakes to proliferation. And indeed, if we lose TGF-beta receptor in the epidermis, there's hyperproliferation going on. You can test it by all sorts of different mechanisms. But it turns out to be counterbalanced by apoptosis. So there's a hyperproliferation, and the epidermis has a backup system that if I see too much proliferation, I'll activate apoptosis, I'll start killing off cells, and the two balance themselves. So if you stand back and look at the epidermis, homeostasis looks perfectly normal. Well, how long can the skin get away with that? Clearly, in terms of cancer, it's starting to give you a view of how you could accumulate mutations over time and have everything seem like it's normal when things aren't really all that normal. And indeed, our skin has about 50% on average. If you've been out in the sun, probably 50% of your cells have mutations in p53 already, even though your skin looks perfectly normal. Frightening. So anyway, I'll give you something more frightening, and this is not a topic for lunch, but I will leave you with this note, and I'll show it quickly, and that is that if you now start to simply apply a tumor-promoting agent or a tumor mutagen, DMBA, known to be presetting the scene for tumorigenesis of the skin, a classical model when it's followed up with a tumor-promoting agent. If you just apply DMBA and don't apply the tumor-promoting agent and take a look at what happens, even though this homeostasis looks perfectly normal, it clearly is not normal. You rapidly progress to squamous cell carcinomas of the skin. And human squamous cell carcinomas of the skin also, as Geraldine Gauche in my laboratory has now shown, also have signs of reduced levels of TGF-beta receptor signaling. And so I would like to really leave you with that, except there's one more very interesting story, and it's just 2 slides to explain the point. And that is that, in fact, when you're missing TGF-beta, we can say, well, the prevailing notion is it's just applying the brakes to the skin, to skin proliferation. We've explained all that. Okay, so there's apoptosis turning on, increased propensity to tumorigenesis. But in fact, some of those tumors metastasize. And we started to look at the cells in culture, and it turns out that there's a cell-autonomous increase in migratory and invasive behavior, losing what was thought to be a receptor that's putting the brakes on growth control. But a proliferating cell isn't a migrating cell. And so what's going on? And so it turns out that in the absence of TGF-beta receptor, there is an enhanced activation of beta-1 integrin and beta-1 integrin signaling. So those 2 tyrosine kinases that are associated with integrins, SARC and FAC, are both upregulated in cells that are just sitting there in a culture dish from the skin of the animals with the loss of TGF-beta. And in fact, in vivo as well, if we use an antibody to activate a beta-1 integrin, or we use antibodies to FAC, we see elevated levels. And if you look in human tumors, you also see elevated levels of FAC, the human squamous cell carcinomas. And in fact, FAC has been knocked out both by Reinhard Fassler's laboratory and also by the laboratory of Lou Reichart and his co-workers, and we've obtained the FAC knockout of the FAC mice and made conditional knockouts and looked at those, as did Margaret Frame. And it turns out that in vivo, what we see is that squamous cell carcinomas show elevated levels of FAC activity, and the FAC conditional knockout skin shows increased tumor resistance. So it's acting exactly the opposite. In the absence of TGF-beta, there's elevated levels of FAC, and there's propensity for squamous cell carcinoma formation. In the absence of FAC, there is a resistance to squamous cell carcinoma formation. And we've studied what happens in the absence of FAC in vitro, and in the absence of FAC, what we've learned, or reversed through activated FAC, that FAC is required for cell spreading, focal adhesion turnover, and migration, but it does not appear to be essential for proliferation or cell survival as it is in fibroblasts. And so, starting at the University of Chicago back in the early 1980s, when we worked on keratin, just as a means of really trying to understand growth and differentiation, of getting markers, we got a bit distracted for a while studying the cytoskeleton. That's been an interest all my career now, and really leading us to degenerative diseases. And then we started a natural segue into the actin and microtubule cytoskeleton and cell-cell junctions. And these people here, Valera Vasukin, Alec Vaezi, Agnes Kobalak, Shrikala Raghavan, who worked on that, had to work in the cold when we were at Chicago until 2002, and then we moved to Rockefeller University. And at the time, 17 people moved with me, but most of these people are new, and I'd just like to give credit to a few people. Pierre Coulombe was the postdoctoral fellow who's now a full professor at Johns Hopkins, has continued to study intermediate filaments and worked out the genetic basis for human EBS. Bob Vassar worked out the genetic basis for mouse EBS. He was my graduate student, is now a full professor in neurobiology at Northwestern University, and he is the person that started transgenic mouse technology in my lab. Tony Latai was the person who did the random mutagenesis studies, and he's the luckiest person in the world, but he's now a professor at Dana-Farber Cancer Institute as a full-fledged MD, PhD. And Kathy Albers did the mutational analysis that guided us to which types of mutations in cells and culture should we put into these mice. Agnes Kobalak did the continued work on alpha-catenin that was started by Valera Vasukin, who's now a professor at the Fred Hutchinson Cancer Center. Agnes just started at University of Southern California. Terry is the person who studied the asymmetric divisions. He's now an assistant professor at Duke. Alec Vaezi is an MD PhD student with me and is trying to be the first successful surgeon who's also a successful cell biologist. I don't know if he's going to make it yet. It's too early in his career, but he does have the ability. Shrikala Raghavan did the beta-1 integrin knockout. She's at Columbia University. Mirna Perez Moreno is just about to be a European colleague. She's going to be joining Erwin Wagner's department at the CNIO in Madrid, and she's the person who did the P120 catenin knockout. Geraldine Gouache, I mentioned already, but she's the person who did the conditional TGF-beta2 receptor knockout, and Marcus Schober did the FAC conditional receptor FAC knockout. And I should mention Amalia Pasoli, who is the electron microscopist in my laboratory, and I think those are the people responsible for the work I talked about today. I know I've kept you here for an extended period of time, and you very patiently sat in your seats, but I've exhausted the video camera, and if there's any questions, I'd be happy to stay and answer them. Thanks.